Hello everyone. In this class, we'll learn about stomach. Stomach and its secretions. The muscle coat of stomach has three layers: an outer longitudinal, middle and uh, middle circular and inner oblique. The gastric epithelium surface shows a number of small openings called gastric pits. There are about 3 to 7 gastric glands open into a single gastric pit. Gastric glands are simple tubular glands located in the mucosa. There are three types of glands: fundic, pyloric, and cardiac. Fundic glands found in the fundus and over the greater part of the body of stomach, and they contain four types of cells: chief cells, parietal cells, enterochromaffin cells, and neck mucous cells. Chief cells secrete pepsinogen and other proteolytic enzymes, whereas parietal cells, are also called as oxyntic cells, secrete HCl and intrinsic factor. Whereas enterochromaffin cell, they secrete histamine, and neck mucous cell, they secrete thin mucus. Pyloric glands. So these are found in the antrum and pyloric regions of the stomach, and they contain two types of cells: the neck mucous cells and G cells. Neck mucous cells, they secrete thin mucus, whereas G cells, they are responsible for release of gastrin. Cardiac glands, they are found around the cardiac orifice and they secrete thin mucus. The different functions of stomach: it mainly acts as a receptor for the food, and it also acts as a temporary storage of the food. It is important for mechanical and chemical digestion of the food, and it also helps in absorption of alcohol, some drugs as well. The intrinsic factor helps in the absorption of vitamin B12, thus helps in the maturation of RBC. And also stomach empties its content at a controlled rate to the duodenum. Composition of gastric juice. Normal gastric juice is around 2.5 liters per day, and the pH is around 1. If you see the composition, 99.5% of is water and 0.5% is solids. Solids include both organic and inorganic components, where organic constituents they constitutes mainly the enzymes, that is pepsinogen, gastric lipase, and gelatinase. The mucus secreted is of two types: the thick insoluble mucus and thin soluble mucus. Thick mucus is mainly secreted by the surface epithelial cells, whereas thin, they are secreted by the neck mucous cells of the pyloric and cardiac glands. And also it contains intrinsic factor. If you see the inorganic components, they mainly made up of hydrochloric acids, sulfates, bicarbonates, sodium, and potassium. Some of the further details about the individual components, like HCl. HCl is necessary for conversion of pepsinogen to pepsin. It has a bacteriostatic action, that is, food contains many bacteria, but they are killed by the gastric HCl, so that the contents of the upper part of the small intestine are bacteriologically sterile. The hydrochloric acid also stimulates the secretion of the GI hormones, and it has a preventive role against the gastric cancer. Persons who suffer from achlorohydria are thus more susceptible to gastric cancer. Pepsinogen. In the presence of HCl, pepsin is very active and it digests the food protein. Thus, the combination of acid and pepsin is called as acid-pepsin mixture, that is APM, and it is a powerful digester of the protein. Mucus. Mucus has the following functions. Especially the thick mucus, it protects the gastric mucosa mechanically from being injured by the corrosive effects of food. And together with bicarbonate ions of the gastric juice, it constitutes the mucosal barrier, which prevents the autodigestion of the gastric mucosa itself by the acid peptic mixture. The soluble mucus makes the food lubricated. Bicarbonates in them act together with the gastric mucus to prevent the autodigestion of the stomach. Then intrinsic factor is needed for the absorption of orally ingested vitamin B12, whereas gastric lipase converts triglycerides into fatty acids and glycerol, whereas gelatinase liquefies gelatin. So this is the protein contained in the connective tissue. Coming to the hydrochloric secretion, the acid formed in the intracellular canaliculus of parietal cell, that is not within the cytoplasm, so that the cell is not damaged due to the high acidity. The intracellular canaliculi extend from apical surface of the parietal cell into the interior, and they contain hydrogen potassium ATPase and chloride channels on the walls. The basolateral membrane on parietal cell in contact with the interstitial fluid and contain chloride bicarbonate exchangers and sodium potassium ATPase. When parietal cells stimulated, the carbon dioxide and water combine to form bicarbonic acid, which splits into hydrogen and bicarbonic acid bicarbonates in the presence of enzyme carbonic anhydrase. The hydrogen is pumped into the lumen of canalicculus with the help of hydrogen potassium ATPase or proton pump, so which is the active transport. The bicarbonate leaves the parietal cells by an antiport in the basolateral membrane of parietal cells that exchange chloride for bicarbonate ions. The bicarbonate ions binds with the sodium to form sodium bicarbonate in the capillary blood. The chloride in the cytoplasm diffuse into the lumen of canalicculus. In the lumen of canalicculus, the H plus combines with the chloride to form hydrochloric acid. This hydrochloric acid reaches the gastric lumen. So this is the diagram showing the production of HCl in the parietal cell, the activated parietal cells. So because of the metabolism, carbon dioxide is produced that combines with water in presence of carbonic anhydrase to form the bicarbonic acid, which forms the H plus ion and bicarbonate ions. So the H plus ions, they are apically, they are secreted into the canalicculus with the help of hydrogen potassium pump. And at the basolateral membrane, the bicarbonates are exchanged with the chloride ions. So the chloride ions, when they are increased intracellularly, they are going to diffuse into the canaliculi, and that is going to combine with H plus ion to going to form the hydrochloric acid, which is secreted into the stomach. At the same time, when the potassium concentration increases in the cell, so that will be exchanged with sodium potassium pump, and when the sodium enters inside the cell, it is going to drag some amount of water also, so which is going to diffuse into the canaliculi, which will be secreted with the hydrochloric acid. For each molecule of HCl formed in the gastric lumen, one molecule of sodium bicarbonate is formed in the blood. Hence, when HCl secretion is increased after meal, the pH of the blood increases, and this is called as postprandial alkaline tide. Some of the inhibitors of HCl or the parietal cell are their somatostatin, the acid chyme. It can have a direct action which inhibits the parietal cells directly, and indirect action will be that it increases the secretion of or production of secretin. Fatty food is going to inhibit the HCl secretion by a release of GIP. So this inhibits the gastric G cells and the parietal cells. Whereas enterogastrone, which is released because of the fatty acid, it is going to inhibit the HCl action of HCl. Then hyperosmotic chyme, which is going to inhibit the secretion and motility reflexly, and prostaglandins, particularly prostaglandin E, is going to inhibit the HCl secretion. Pepsinogen secretion. Pepsinogen is an inactive precursor of pepsin. It is secreted by chief cells. Pepsinogen has no digestive activity. However, as soon as it comes in contact with HCl, it is activated to form active pepsin. Pepsin functions as an active proteolytic enzyme in high acid medium. That is, the optimum pH required for its action is 1.8 to 3.5. But above the pH of 5, it has no proteolytic activity and becomes completely inactivated in a short time. Secretion of intrinsic factor. The intrinsic factor is secreted by parietal cells of gastric mucosa along with the secretion of HCl. The intrinsic factor is essential for absorption of vitamin B12. It forms a complex with vitamin B12 which is carried to the terminal ileum where the vitamin is absorbed. When the acid producing parietal cells of the stomach are destroyed, so which frequently occurs in chronic gastritis, the person develops not only achlorohydria, that is lack of stomach acid secretion, but often also pernicious anemia because of failure of maturation of red cells in the absence of vitamin B12, that stimulation of the bone marrow. Regulation of secretion of gastric juice. Gastric secretion is accurately synchronized with the need for gastric juice. The mechanisms regulating the gastric secretion include neural control and chemical control. Neural control. Neural control over the gastric gland is exerted by the local enteric plexuses involving cholinergic neurons and impulses coming from central nervous system via vagal innervation. So that forms the extrinsic nervous system. Vagal stimulation increases the secretion of HCl by parietal cells and pepsin by chief cells. The vagal stimulation increases the H plus ion secretion by direct and indirect path. In the direct path, the vagus nerve fibers innervating the parietal cells stimulates H plus secretion by releasing acetylcholine, which acts on these cells through the M3 muscarinic receptors, that the molecular mechanism to increase the intracellular calcium on parietal cells, and it causes the direct stimulation of the gastric juice. In addition, acetylcholine also potentiates the effect of histamine on H2 receptors on the parietal cells. In the indirect pathway, the vagus innervates G cells and stimulates the release of gastrin into circulation through gastrin releasing peptide. The gastrin in turn stimulates the H plus secretion. The acetylcholine released on the vagal stimulation also acts on enterochromaffin cells, so which release the histamine. Histamine increases the H plus secretion by acting on H2 receptor on parietal cells, but the relative importance of acetylcholine stimulating their secretion is unsettled. Peptic cells are stimulated by acetylcholine released from vagus nerve or from the gastric enteric nervous plexuses. Chemical control. So different chemicals play important role of gastrin as it is a GI hormone produced by G cells located in the pylori glands, the most known powerful stimulant of HCl secretion. Presence of the products of the protein digestion in the stomach acts on G cells to release gastrin. It stimulates secretory activity of the parietal cell and chief cells. The actions of gastrin are: gastrin stimulates the secretion of histamine from enterochromaffin cells. Histamine stimulates the hydrochloric acid secretion, and this is the principal way by which gastrin stimulates acid secretion. Gastrin also stimulates hydrochloric secretion by acting on the gastrin receptor in the parietal cell. This increases the intracellular calcium. To a lesser extent, it stimulates the chief cells to secrete pepsin. Coming to the role of histamine released from enterochromaffin cells. Enterochromaffin-like cells bear both gastrin receptor and acetylcholine receptors. They release histamine in response to both circulating gastrin as well as acetylcholine released by the vagus fibers. The histamine release stimulates hydrochloric acid secretion from parietal cells by acting on H2 receptors. Histamine bind to H2 receptors via G proteins, that is stimulatory, and this increases the adenyl cyclase activity and intracellular cyclic AMP. Increase in the intracellular cyclic AMP and calcium act via protein kinases to increase the transport of H plus ions into the gastric lumen by hydrogen potassium ATPase. Then role of somatostatin. Somatostatin is secreted by D cells in the gastric glands and they inhibit gastric acid secretion. Whereas prostaglandin E2 acts via G proteins, that is they're mainly inhibitory and decrease the adenyl cyclase activity and intracellular cyclic AMP, and this is going to inhibit the acid secretion. The low pH in the stomach inhibits the secretion of H plus ions by parietal cells by negative feedback mechanism. The intestinal influences like fats, carbohydrates, and acid in the duodenum inhibit gastric acid and pepsin secretion via neural and hormonal mechanisms. The rate of secretion of pepsinogen is strongly influenced by the amount of acid in the stomach. Acids elicits additional enteric nervous signals to the peptic cells. Emotional responses, psychic states have effect on gastric secretion and motility that are principally mediated via the vagi. Anger and motility are associated with hypersecretion of the gastric mucosa, whereas fear and depression decrease gastric secretion and blood flow and inhibits gastric motility. So this is the molecular mechanism, how different chemicals they are going to act on. Acetylcholine, it is going to act on M3 receptors. Gastrin, it is going to act on the gastrin receptors where they are going to increase the intracellular calcium. And whereas histamine and prostaglandins, they are going to act on the G receptors, whereas histamine is going to stimulate the production of HCl, whereas prostaglandins, they are going to inhibit the acid secretion by altering the levels of cyclic AMP. Increase cyclic AMP, increase the activity of the protein kinase with the calcium, so that is going to increase the hydrogen potassium activity to increase the production of HCl. Phases of gastric secretion and their regulation. The phases of gastric secretion can be cephalic phase, gastric phase, and intestinal phase. Cephalic phase of gastric secretion occurs before the entry of food in the stomach. The secretion is initiated by sight, smell, and thought of the taste of the food. The cephalic phase is under neural control and it is mediated through the vagus. The 20% of the total secretion occurs in this phase. Gastric phase of gastric secretion occurs when food enters the stomach. 70% of the total acid secretion occurs in this phase, and it is mediated by neural and hormonal mechanism. The neural mechanism is mediated by the distension of the stomach by food, which is going to stimulate gastric juice secretion, which can occur through the local myenteric and vagovagal reflexes. Then chemical or hormonal control, where products of partial digestion stimulate the secretion of gastrin. Gastrin enters the circulation and stimulates the gastric glands. Intestinal phase of gastric secretion begins as the chyme begins to empty from the stomach into duodenum. This accounts for 10% of the gastric juice secreted. Initially, it occurs when the early phase of the intestinal phase. The secretion is increased and later it is inhibited. So this inhibition results from the following influences. It occurs mainly because of the enterogastric reflex and hormonal mechanism. Enterogastric reflex is a reflex mechanism mediated through mesenteric nervous system as well as through extrinsic sympathetic system and vagus nerve. It is initiated by distension of small intestine in the presence of acids or protein breakdown products in the upper intestine and irritation of the mucosa, where by the reflex mechanism they will be inhibiting the production of HCl from the stomach. Hormonal mechanism, presence of acid, fat, protein breakdown products and hypoosmotic or hyperosmotic fluids and any irritating factors in the upper small intestine causes the release of hormones like CCK, GIP, VIP and somatostatin, which inhibits gastric juice secretion. So these are the different phases of gastric juice secretion: cephalic phase, gastric phase and intestinal phase. The cephalic phase is mainly the nervous regulation, where the sight, smell, thought and hearing of the food by the conditioned reflex they can increase the HCl or gastric juice production. And when the food enters into the mouth, it can lead to the unconditioned reflex and it can increase the HCl production. Where gastric phase is when food enters inside the stomach, that increases the vagal secretion that leads to the increased gastric juice secretion. Whereas the intestinal phase, and it can act both by the neural mechanism as well as hormonal mechanism. Intestinal phase is mostly the hormonal when the chyme enters into the upper part of the intestine. So there will be release of various gastrointestinal hormones like secretin, CCKPZ, somatostatin, GIP and VIP. So they are going to lead to the inhibition of gastric juice into the later part, and this is called as the enterogastric reflex. Whereas in the initial part, the 10% of the gastric juice is caused by the production of the gastrin. Sham feeding. It is an experimental procedure devised by the Pavlov to demonstrate the regulation of gastric secretion. Procedure is where a hole is made in the neck of anesthetized dog. The cut ends are drawn out through the hole in the neck. When the dog eats the food, it comes out through the cut end of the esophagus. But the dog has the satisfaction of eating the food. So this experimental procedure is supported by the preparation of Pavlov's pouch with a fistula from the stomach. The fistula opens to the exterior and it is used to observe the gastric secretion. It is useful to demonstrate the secretion of gastric juice during cephalic phase. In the same animal, after vagotomy, the sham feeding does not induce gastric secretion. It proves the role of vagus nerve during the cephalic phase. So this is in brief about the stomach and the phases of gastric juice secretion. Thank you.